Hello, everyone, and welcome to today's live broadcast, Measurement of Pesticides in Cannabis, presented by Jeffrey Dahl, Application Scientist at Shimadzu. I'm Alexis Krause of Labroots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by Labroots. Labroots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom center of your screen, or use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Dahl. I will now turn the presentation over to him.
Hello and welcome to this presentation. It's my pleasure to present for you. And the topic of today's talk is measuring pesticides in cannabis. And the techniques that we're going to talk about are industry standard for measuring pesticides in food and vegetables, and now we're applying those same techniques with the appropriate modifications to cannabis. So as everyone knows, the cannabis industry in the United States has really taken off in the last few years, and many states have rolled out either medical and recreational programs. And in support of that, testing is required for a variety of analytes in the sample. Potency measurements are used to determine the amounts of the cannabinoids in the sample, which could vary from strain to strain. Residual solvent testing is required for prepared products to make sure that any residual processing chemicals, the solvents that were used to extract the cannabinoids, are no longer present, and those would be used for cannabinoid concentrates. Microbiology testing is used to make sure that the products haven't spoiled. Heavy metals testing is used to make sure that the plants haven't become contaminated with different types of harmful metals such as mercury or arsenic. Terpene measurements are used to profile the flavor and the chemical attributes of the strain. And finally, pesticide testing is required to make sure that the products aren't going to cause consumers any health problems. Cannabis is a high dollar value crop, and there's a high risk of mold or insect attack, especially when it's grown indoors because of the moisture levels, but also outdoors since it's more open to the environment and it's easy for insects to come in. And obviously once an infestation takes hold, either of insects or mold, the crop is ruined and a lot of money is wasted. So farmers, conventional farmers who grow fruits and vegetables, but also cannabis farmers want to protect their crops, and frequently pesticides are applied in order to prevent that. However, from the consumer point of view, pesticides really haven't been tested in cannabis and they've not been approved for use for cannabis. So from the consumer standpoint, there's a really unknown variable, which is how will smoking cannabis that's been sprayed with pesticides affect the consumer. So various states have adopted different regulations for pesticide testing in cannabis. Colorado, Nevada, Oregon, Washington, California have all adopted regulations for cannabis testing, and in each of those states and others, cannabis testing labs have been established and accredited by the states, and they've already started testing products in many states. And the rules pertaining to that have been in have been rolled out in phases. First, the regulations are proposed, then they're adopted, and then finally enforced. And each of those states, however, has adopted a slightly different monitoring list. And so this, for us, represents a continuously changing problem. But luckily, due to our methods, we're able to add in and change the methods to adopt the state requirements. So, for example, these 4 documents show the state monitoring lists from Oregon State, Nevada, Washington State, and Colorado. California has also come out with their list, as well as Canada and many other states as well. So LCMS is the main technique that we use for pesticides because it's very robust. It can detect the widest range of analytes, and it'll be the focus of this presentation today. However, depending on which pesticide requirements a lab is following, they may need to add GCMS as well. And GCMS is particularly well suited for certain analytes, typically the older pesticides, which are not used much anymore, but depending on the state's requirements, the GCMS may be needed to meet all of the state's requirements, but it varies from state to state. So in our present method, which is an LCMS-based method, we first start out with a representative test portion. And the typical test portion is 1 or 1.5 grams of dried flour. The first step of the sample preparation is to homogenize the sample. And what this does is it allows us to select a representative amount as well as grind up the plant material, which will allow the pesticides to be extracted more easily. If the pesticide was incorporated into the plant matrix and we didn't grind up the sample, then it might happen that the pesticides could be in the product but not detected. So it's important to grind up the sample to release the pesticides from the plant matrix. So for grinding, we investigated several different techniques for grinding. And our criteria to get the best result was to find out the grinding method which gives us a uniform, homogeneous, and small particle size, but while having high throughput and being easy to use. So displayed here are the several methods of grinding that we investigated. The herb grinder is the typical grinding. We tested a handheld homogenizer, a coffee grinder, a tissuelizer, which is consists of Teflon grinding jars with a grinding ball inside, the genome grinder, the magic bullet, and a flax mill. So we found that the herb grinder gave a particle size that was too coarse. It was very uniform, but when the particles aren't small enough, the pesticide extraction might be inefficient. We found the handheld homogenizer became clogged very easily, and it was time-consuming to clean. The coffee grinder gave a very excellent, very fine particle size and homogeneous, but typically was better suited for larger sample volumes. So depending on the state's rules, this might be a suitable option. The tissuelizer gave also a very fine powder, but this is more difficult to clean since the grinding jars have to be cleaned between each sample. Finally, we found that the genome grinder was the best choice because it gives a very fine homogeneous particle size, but also has very high throughput since it doesn't require cleaning the grinding jar with every sample. So for this presentation, we used the genome grinder. So after grinding the sample, we need to extract the pesticides with a solvent extraction. And we found after testing a variety of techniques that a solvent extraction is the best. And our solvent extraction starts with 1 gram of powdered sample, and we extract that with acidified acetonitrile with shaking and homogenization. And sonication also is a good way to help extract pesticides. This solvent, after extracting the pesticides out, will spin the sample down in a centrifuge, and this will allow the plant material that remains to pellet in the bottom, and then we'll take the supernatant, which is the acetonitrile extract, to the next stage of the analysis. And this slide shows an example of how that looks. On the left, you see the flower sample. In the middle, after being ground up in the Geno grinder with 2 steel balls. And then finally, on the right, is the solvent extracted sample. Now, we could inject this directly into the instrument. However, in order to keep the instrument cleaner, we like to do a cleanup step. And the cleanup step involves taking the sample extract and subjecting it to a cleanup treatment with a material which is called dispersive SPE. And SPE is the solid phase extraction. And this consists of several ingredients which are designed to adsorb the matrix, that is the undesired compounds in the sample. In this case, we tested several different formulas that consist of a PSA, that's a primary secondary amine, GCBE is a graphitized carbon black, and C18 is the octadecylsilane material, and in different ratios. So we found that if the cleanup was too strong, such as Formula 2, we lose too many pesticides. So we selected Formula 1, which has a lower amount of carbon, and that gives us good recovery of pesticides while removing a lot of the matrix components that we don't want. So this is a fairly fast cleanup, and it's only needed to shake the sample with this powder and then spin it down again and take the supernatant and directly inject for analysis. For our present analysis, we use the LCMS 8050 triple quadrupole mass spectrometer. We make several different models depending on how low of a detection limits are required for a lab's needs. So we also have a more sensitive LCMS 8060 and also several more entry-level models. And this is the instrument used in the present analysis. So our sample is injected onto an LC column, and our separation is performed, and the pesticides will elute with an expected retention time, and an example chromatogram is shown in this figure. And for each analyte, we detect its presence by using the machine settings in MRM mode. And we've optimized the MRMs for each analyte to get the maximum signal. And we've also adjusted the chromatography to give us good separation between the interfering components and the analytes of interest. So using this method, we prepared calibration curves. This is just a representative selection of several important analytes down to the typical required detection limits in various states. And we're showing linear results for all these analytes. Next, to further validate our method, we analyze triplicate samples for our calibration curves. And this is showing the RSD of those triplicates in a wide concentration range to be well below 20%. So it's showing that we have very good reproducibility. And next, we're showing the replicates for the QC samples. The quality control samples are used to verify the method's accuracy, and what we're showing here is for the 3 different spiking levels of QCs, just showing the 50 nanogram per gram level, and we're showing the RSDs well below 20% in 3 different strains. Next, we show the recovery, and the recovery is important so that we know that we are not losing the analyte in the extraction process. And for nearly all the compounds, we have recovery between 70 and 120%. Next, in order to determine whether or not there might be any strain-specific problems with the analysis, we analyze several different, all the compounds in several different strains. And you can see here that in each of those strains we get a similar result, and that indicates that the method performance is very good even using different strains of cannabis. And just if anyone's interested, for this analysis we used one strain was Dutch Treat, one strain was Orange Kush, and there was a third strain that was composed of a composite of several different strains. So this is our analyte list for the present method. And this is well below the required detection limits for various states. And this list actually is much longer than is required for California, Oregon, and even Canada. So next, we've taken our validated method and run real samples that we obtained from the market in a blind selection process. And in this case, we've selected 27 dried flower samples from Washington State and used our method to detect how many samples actually contain pesticides. And this analysis was done about 12 or 16 months ago, and we have found actually a somewhat higher than expected rate of positives from some of these samples. Some of the more recent ones are less positive, but for this sample set, we detected pesticides in about half of the samples. The most commonly detected was piperenol butoxide, and this is a pesticide that's used in combination actually with the pyrethrins, which don't often survive due to oxidation in air. If we remove those, then actually the positive rate is only about 25%. And we can also see several pesticides we detected that were not on the state monitoring list. So next, we show the results for the cannabis concentrates. So cannabis concentrates, of course, can be prepared from cannabis material using several different methods. Some of them are CO2 extractions, some of them are solvent extractions, and some of them are using propane or butane extractions. And depending on the extraction conditions, these may be carrying over the pesticides that were originally in the plant material into the finished product. And in fact, due to the concentrating effect of the solvent, not only does it concentrate the desired cannabinoids, but it might also concentrate the pesticides as well. So we selected a range of concentrates. The top row in the pictures are showing shatters, and the middle row is sort of waxes, and the bottom row are more oils. On the right side of the screen is a vape pen. And we've tested quite a wide range of these types of concentrates. We also tested a wide range of other types of concentrates, and there's various pills and powders and oils. This is a representative selection of what we've tested. So the first step in the analysis is to dissolve the sample. So we found after doing a dissolution test that methanol was the most effective solvent. And for nearly all of the different types of concentrates, we found methanol was very good at dissolving all of the cannabinoids and all of the solid material. And so we use this as the first step of our pesticide analysis for concentrates. And really the cannabis concentrates are much cleaner already than the dry flower material, so we don't use a cleanup procedure for the concentrates, simply a dilute and shoot. We run all the samples with a internal standard, and we also carried out a single point standard addition experiment to verify the absence of matrix effects on these samples. So shown in the picture are a batch of 50 different concentrates. You can see a wide range of color that we find depending on how much chlorophyll remains in the sample, as well as the other co-extracted components of the sample. So after extraction and filtering, and we run by the same LCMS analysis, we can show the same validated method for cannabis concentrates as well. And on this slide, we're showing the single point standard addition results, showing that we are detecting spiked pesticides as we expected for most of the analytes. And on this slide, we're showing the results of our market survey for cannabis concentrates. In this case, we use 48 samples from the Denver market, and we're finding a very high positivity rate. And we think this is due to the fact that in the cannabis concentrates, the pesticides are actually concentrated more than they were in the flower, and then by that fact, they are going to be above the required action limit in each state. So it may be more likely for cannabis concentrates to test positive for pesticides than the flower material. So in this case, we're showing over 80% positive rate. And again, pipernobutyl oxide, pyrethrins are the most common. Also myclobutanol and some of the neonicotinoids are also detected as well. And for comparison, in Spokane, we looked at about 100 samples, and this is, again, a similar selection of cannabis concentrates. And we found a similar positive detection rate that we found in Denver. That's over 80% testing positive, and again, the same pesticides testing positive that we found in Denver. And I want to call attention especially to several important compounds on this list, which are not required to be monitored by the states, in particular carbendazim. So carbendazim is an antifungal that made the headlines a few years ago, being found in orange juice. And it has antifungal activity, so it's no surprise to find this on cannabis. But it's not actually required to be monitored in any of the states that we visited, or in Canada either. But we found it in about 10% of our samples. So this could reflect a trend among growers to switch to unlisted pesticides. And finally, to show the method robustness, over a 2-week period and almost 1,000 injections of samples, we show that the instrument performance is very good. We can measure that by retention time, by peak width, ion ratio, and signal intensity. And this shows that this method is well suited to high throughput and also long-term analysis of pesticides in cannabis, even with these very complicated samples. So to conclude, we found that pesticides were used in cannabis and fairly widely. We found that our method gave a very good result in terms of being efficient to use and sensitive to detect low levels of pesticides. We found that the cannabis concentrates could be analyzed by a simple dilute and shoot method, and we found very good LCMS robust performance. And we also found that there could be growers switching to pesticides that are not on any of the state monitoring lists. So it's important for states to keep aware of new pesticides that are not currently being monitored. So in conclusion, I want to thank our collaborators. All the work that we've done has been in collaboration with state-certified laboratories since the product is federally illegal. So especially we worked with members of Trace Analytics in Spokane, Washington, as well as members from other organizations as well, providing consumables. And we also want to thank our collaborators at Nordic. And I want to thank all of you in the audience for your attention, and I'd be happy to answer any questions you may have.
Thank you, Dr. Dahl, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. We'll answer as many of your questions as we have time for. So let's get started. Our first question is, can a smaller sample amount be used? And how are the samples collected?
Well, that's a good question. In the cannabis industry, there's strong pressure to use smaller samples because the product is very valuable. But on the other hand, we have to make sure that the sample is representative. And so typically what the states require is that sample collector represents the lab and collects a sample in a statistically sound sampling procedure. And usually that means going to a production lot and selecting a small subsample from a randomly assigned part of the batch. So if we had a 15-pound batch, the collector will select a small sample from several different places throughout the batch to ensure that a representative sample is selected. And depending on the state's rules, the sample might range from 10 or 20 grams, for example. And from that larger sample, we would then select an even smaller subsample. So in the presentation, we used a 1-gram sample for the pesticide analysis. In principle, there's nothing stopping us from using a smaller sample because the instruments are able to detect pesticides from a smaller amount. However, to get a good representativeness, typically we would use a 1-gram subsample.
And our next question is, can a test for potency be done in the same analysis as the pesticides?
So the potency measurement is quite a different measurement than the pesticides. The instrumentation is capable of doing both pesticides and potency, but because of the large difference in concentration, we usually would do a different dilution to do the potency measurement. So the pesticides might be present in concentrations as low as 5 or 10 parts per billion, whereas the cannabinoids might be in concentrations that are more of, several percent up to 20%. And so typically, even though the instrumentation could make the measurement, we usually need to dilute the sample more for the potency measurement. And due to some other factors such as the ease of calibration, we usually recommend running potency by an LCUV and then dedicating the LCMS towards the pesticide measurement.
And Dr. Dahl, it looks like we have time for one more question. Can you detect pesticides that are not on the state-required monitoring list?
So with our measurement techniques, the typical workflow is to use the MRM mode for detection. And in MRM mode, we're typically starting with a targeted pesticide list. And in our presentation, we are covering a larger list of compounds than is required by any of the states. And we can certainly add many more pesticides to those monitoring lists. And in fact, we found pesticides in cannabis samples that were not actually required to be measured by the states. And this could reflect growers switching to different pesticides. So we certainly can expand the monitoring list beyond what the states require.
I would like to once again thank Dr. Dahl for his presentation. I would also like to thank Collaborators for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through June of 2018. You will receive an email from Collaborators letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.